Уважаемые коллеги, мне сегодня очень приятно видеть нашу любимую аудиторию, заполненной большим количеством слушателей. Мне очень приятно, что здесь много молодых людей, помимо именитых профессоров. И сегодня здесь выступает профессор Гилберт, который, наверное, многим из вас известен лучше, чем мне, поскольку я далек от эволюционной биологии развития. Это специалист в области биологии развития, который, насколько я знаю, активно работает в нескольких направлениях, в частности, в эволюционной биологии развития и в экологической биологии развития. И его работы известны достаточно широко во всем мире, и я уверен, что сегодняшняя лекция, которую он прочтет здесь, и те лекции, которые он будет читать в рамках фестиваля науки, будут интересны для тех, кто их прослушает, поэтому я не буду больше времени занимать, а предоставлю слово профессору Гилберту. Спасибо большое.
I will make some introduction, scientific introduction in Russian.
Sure.
Разрешите мне сделать, так скажем, научное вступление для студентов, чтобы было полегче ориентироваться в лекции, которые будут проходить на английском языке. Я сейчас очень коротко по-русски обрисую проблему. Профессор Гилберт будет рассказывать о проблеме эволюционных новшеств. Что такое эволюционные новшества? Это признак, которого не было никогда раньше. В палеонтологической летописи мы не можем его найти. Обычно эволюцию признака можно проследить, когда мы смотрим на палеонтологическую летопись, когда мы смотрим на близкие виды и другие таксономические группы. Но бывает так, что вот в эволюции впервые возник какой-то признак. И это на самом деле большая проблема, ведь признак может эволюировать под действием естественного отбора только тогда, когда у него есть функциональная нагрузка. А когда он только что появился, он не может работать, он не может функционировать. Соответственно, его носители не получают никакого преимущества, и, соответственно, эволюция под действием естественного отбора невозможна. Откуда же такие признаки берутся? И вот выясняется, что признаки такие появляются благодаря изменениям, происходящим в нормальном развитии, эволюционным изменениям. И оказывается очень удобная модель для изучения таких вот появления эволюционных новшеств – это черепаха, точнее панцирь черепахи. И профессор Гилберт со своей группой как раз занимается изучением развития панциря черепахи, чтобы понять, как появился этот эволюционно новый признак, которого у предков черепах когда-то не было. Все появляется когда-то впервые. Спасибо.
Okay, just going to stay back here. Thank you very much. It is a great pleasure to be here today. I'll be talking about a question which involves both developmental biology and evolutionary biology. The question is, how does newness come into the world? And I'll be using as an example the turtle shell. And I couldn't resist putting up this slide because this is a children's book. And so this question of how does the turtle get its shell is a fundamental question. It is a question that 9-year-olds ask. It isn't like, what does the L7 protein of the ribosome bind to? This is, a critical question. How does the turtle get its shell? I want to also talk about newness in one of two contexts. There are different levels of novelty, different levels of newness. I will be talking about the second. The first level of newness is, how do you get a new cell? how do you get something that hasn't existed before on the cellular level? And to get a new cell probably means changing the relationships of enhancers and transcription factors in the nucleus. There are several areas of investigation going on right now. They are preliminary, they are fascinating, and they are trying to answer the question of how do you get fundamental newness, a new cell type. For instance, Günter Wagner's lab at Yale University is working on the question of how do you get the uterine cell, the decidual cell of the uterus, which responds to progesterone so that the pregnancy of mammals can be internal. How do you get a cell type that forms the uterus? And what he's finding is that this new cell type probably originated by viral infection, that retroviruses seem to position themselves with progesterone responsive enhancer elements next to certain genes. And that caused, if you will, a rewiring of the circuits of the cell, allowing certain genes to become responsive to progesterone and allowing the mammalian uterus to evolve. Another group centered in Michael Levine's laboratory at University of California, Berkeley, is looking at how do vertebrates get the neural crest cell. The neural crest cell is the cell that forms our sympathetic neural lineages. It forms the pigment cells of our body. It forms our facial cartilage. It forms our facial bones. It forms the anterior part of our skull. This is a new cell type. It didn't exist in invertebrates. And so he is finding that the twist gene, a transcription factor, is absolutely critical in forming the neural crest. It involves cell migration. It involves getting out of epithelial layers. And this cell type, if you add twist to a tunicate larva, such that it is expressed in certain cell types, which are melanocyte, they're pigment cells, you actually can get the rudiments of a neural crest cell. So there are some people who are working at the level of newness. How do you get a new cell type? I am looking at the level of newness of how do you get a new organ or a new anatomical structure made out of cell types that already exist. So I'm going to look at how does the turtle get its shell. These are bony elements. These are elements that are found in all vertebrates, but they're organized in a new way. And so this is the type of newness I want to look at, which is how do you take existing things and put them together in new arrangements? And Carl Ernst von Baer in 1834 said, but the developmental scheme in the case of turtles must be modified in quite a unique fashion, which on closer examination of their development makes us very interested. But he couldn't go further. Heinrich Rathke did go a bit further and look at what was happening during turtle development, but he could not explain how it happened. He could explain that the thing that makes a turtle are its ribs. The ribs of a turtle do not form a rib cage. They don't go around the lungs like our ribs do. The ribs of the turtle go laterally into the dermis of the skin, and they help form the shell, the upper shell of the turtle. So you can see over here, these are ribs. And the turtle shell is made, there's a carapace here, the upper shell, and a plastron, the lower shell, and they're connected by a bridge which is made from the plastron cells. Now, in America, the creationists say that you can't explain turtle origins because they came on day 5 of creation. And there's actually a museum, and they say here that turtles appear abruptly in the fossil record. That's the only thing turtles ever did abruptly. This is a problem not easily explainable by the evolutionist, but is easily explained in the biblical creation model. And my job is actually to say, actually, evolutionary biology can explain the origin of turtles. Okay, this is Creation Magazine here. It gets very personal because they say over here that the title of the paper by Leeson and Gilbert summed up the evolutionary conflict perfectly. The new discovery of the beautifully preserved fossil Odontocaelus produces more questions than it answers, reopening questions of turtle origin, shell evolution, and original paleoecology. So yes, we are being honest. We're saying we don't know the answer. And of course they say that's because they arrived only 45,000 years ago, 4,500 years ago. So, but it got personal putting my name in this. Okay, usually when we discuss how newness enters the world, we talk about evolution by natural selection. And this is kind of Darwin and Wallace's idea that there's variation within a population, that there's competition for survival, with most of the organisms dying before they reproduce. There's a survival of those most fit for the environment, and the offspring are from the survivors. And in the 1920s, it was added for the modern synthesis, the beginning of the modern synthesis, the offspring inherit the genes that made their parents fit for the environment. So this is evolution by natural selection. It works. It's been tested for over 100 years. It's been found accurate in every case. But this is not the whole of evolution. This is the survival of the fittest. What other evolutionary biologists were interested in, this just shows that, yes, there's variation in the population. These are some of the people who've worked in my lab. Death is very important, and if death is not around, one aphid can give rise, and it's only one because they're parthenogenetic, they don't need males, 524 trillion aphids in a year. The housefly, 2 times 10 to the 20th in a hot summer. Staphylococcus aureus, the bacterium, can cover the whole planet 2 meters deep in 48 hours. Okay, death is critical. Death is not always random, it can be selective, and here we have the beak of the finch that in Geospes fortis, one of the species of Darwin's finches, during a drought of 1976 to 1978, the population went down, but those that survived had bigger beaks, and that allowed them to crush nuts that the other finches could not. We know that the survivors have offspring who inherit the genes that made their parents fit. So Darwin's finches, the beak became larger. The current mosquito has esterase genes copied over 200 times. These are the genes that degrade pesticides. Similarly, the housefly and the cotton budworm moth have mutations in the receptors that bind the insecticide. Homo sapiens, we're still evolving. We have genes that have evolved against malaria. Selection of hemoglobin S, the Duffy blood group substance, absence, and also a specific allele of G6PD. So we are evolving. Bacteria, resistance to penicillin, streptomycin, and so forth. So yes, we have survival of the fittest. But that isn't the whole story. Darwin's friend and colleague, Thomas Huxley, put it this way. He said, you not only need the survival of the fittest, you need a theory of the arrival of the fittest. How do you get the variation that nature selects? And he was talking in the 1890s, saying evolution is not a speculation but a fact, and it takes place by epigenesis, by embryonic development. Darwin would probably have finished the sentence, it takes place by natural selection. Darwin and Huxley were looking at different aspects of evolution. Now, the reason Huxley could say this, and I talked about this in the first lecture that I gave, the reason Huxley could say that evolution takes place by epigenesis were the experiments and the observations of Alexander Kovalevsky and Ilya Mechnikov, who went from Russia to Naples to do their research. And in the 1860s, 1865, they discovered that the tunicate, the larva of the tunicate and the larva of Amphioxus had a dorsal neural tube, it had a notochord, and they had pharyngeal arches and pharyngeal pouches that came from the same germ layers as those of frog and fish neural tube, notochord, and pharyngeal arches. So they were able to say that there was continuity in the animal kingdom between invertebrates and vertebrates, something that hadn't been done before. Darwin was thrilled. He wrote in 1874, when he was writing Descent of Man, here we have evidence for the continuity of the entire animal kingdom. And continuity and community of embryonic structure shows continuity and community of phylogenetic descent. That evolution could be looked at, as von Baer said it could be, by looking at the early stages of development and see how you get changes in anatomy by changes in embryology, by changes in development. So we have this new model, a model that says not only evolution by natural selection, but evolution by changes in development. Now, we had no model for that. And in 1942, another Huxley, this is Julian Huxley, here's Thomas Huxley, this is an ontogenetic series, that's Julian when he got older. A study of the effects of genes during development is as essential for an understanding of evolution as are the study of mutation and that of selection. That a study of the effects of genes during development. Well, this was 1942. We didn't have such a thing. It was only in the 1970s that you could start studying the effects of genes in development. And Susuno Ono produced a theory of gene duplications being important. Alan Wilson and Mary Claire King put forth a model, which I'll mention later, where there are two sets of genes. One which is involved in making proteins, and the other which is involved in constructing embryos. And they looked at chimpanzees and humans. And then Francois Jacob said, all evolution really is about tinkering with existing genes. That genes very rarely change. What we're talking about is changes in the time of gene expression, changes in the amount of gene expression, changes in the cells that the genes are expressed in, and sometimes changes in the structure of the genes. But he didn't think the structure of the genes, the actual gene sequence of the protein encoding region of the gene, changed that much. And the data were actually from Wilson and King. And I show this slide here. This is from the Wilson and King paper. The picture is not from the Wilson and King paper. This is the former president of the United States and with one of his co-stars in a particular movie. But Wilson and King, 1975, said small differences in the time of activation or in the level of activity of a single gene could in principle influence considerably the systems controlling embryonic development. The organismal differences between chimpanzees and humans would then result chiefly from genetic changes in a few regulatory systems, while amino acid substitutions in general, the coding sequence of the gene, would rarely be a key factor in major adaptive shifts. So they're saying, look for things like changes in hemoglobin to be important. Look for changes in the regulation of hemoglobin. Don't look for a particular mitotic gene to be important. Look for changes in the amount of this gene, or in the timing of its expression, or in the cell it's expressed in. So what we now have is this new area Evolutionary Developmental Biology, EVO-DEVO, which says that evolution results from inherited changes in development, and that developmental trajectories can bias which changes are more probable than others. This is called developmental constraints. It also postulates that a more comprehensive theory of evolution results when the population genetic approach of evolutionary change, which is based on allele frequency, is supplemented with a developmental genetic model of body construction that's based on gene expression. So you take natural selection, gene frequency, and you put it together with developmental biology, gene expression. This is a relatively new science. The first division of this was in the year 2000. The first two journals were in 2000. And the first society, the European Society for Evolutionary Developmental Biology, was 2006. So it's a new field. However, if you want to look at the precursors of it, you'll go back to Ilya Mechnikov and Alexander Kovalevsky. So one of the things that evolutionary development says about newness is this. And this is from a paper by Hughes and Kaufman, and they were looking at all the arthropods and saying, where did these new adaptations come from? And they said, to answer this question of arthropod adaptations by invoking natural selection is correct, but insufficient. The fangs of the centipede and the claws of a lobster accord these organisms a fitness advantage. However, the crux of the mystery is this: from what developmental genetic change did these novelties, did these new things arise in the first place? Now, as I mentioned, and that's Mother Nature tinkering, amphipod or something over there. There are generally only 4 ways of tinkering. This is very good. You don't have to do it all the time. All sorts of new things. Just 4 ways. Changing the time of gene expression, the place of gene expression, the amount of gene expression, and the kind of gene being expressed. And again, I talked about this in an earlier lecture. If you want to know how the duck got its webbed feet, so ducks have webbed feet, chickens and humans do not, and that's because in chickens and humans, there's cell death in the webbing between our fingers, between our toes. In the duck, there's very little cell death. This is tunnel staining for apoptosis. So the webbing remains. Why is it that these cells die in the chicken but not in the duck? And if you look at these chemicals which are telling the cells to die, the BMPs, you find that they're expressed in both the chicken and the duck webbing. Why are the cells in the duck not dying? They're being told to die. There's gene expression differences. This is the expression of the gene for gremlin. Gremlin is an inhibitor of BMP. So there's gremlin in the duck webbing, but not in the chicken webbing, not in the human webbing. And as a result, the BMPs here cannot work. They're being inhibited by gremlin. So the BMPs can't work. These cells are not being told to die. You keep the web. Here, there's no gremlin in the web. BMP is active. It tells the cells to die, and you have toes instead of a webbed foot. So the place of gene expression, in this case, the place of gremlin expression, is telling the cells what to do, and you get an anatomical change due to changes in development. And you can show this by experiments. You can take a bead that has gremlin on it, and it will preserve the webbing. If you take a bead that has just salt on it, saline solution, the webbing disappears. So gremlin, the place of gene expression, is really important. The time of development, the timing of development is very important. Here we have the ratio of brain to body, and you see it growing at a certain level, and in the human, it keeps growing for over 2 years. The brain keeps on developing at the fetal level, keeps on making more and more neurons, where the chimpanzee basically stays the same. It keeps up the same, it slows down its brain growth relative to body for the first 2 or 3 years, but not the human. It keeps its mitosis going. And we know some of the genes that are involved in this. The GADD45G gene, the SRGAP2 gene, these are genes that normally inhibit mitosis in the brain. They stop cell division in the brain. And we have evolved inhibitors to those genes so that these genes are stopped, these genes don't function, and the brain keeps on growing. So changing the place of gene expression is important. Changing the time of gene expression is important. Heterometry is changing the amount of gene expression. These are Darwin's finches. Here is one species of finch with a broad beak which can crack nuts. Here we have another species of finch with a narrow beak which can get insects out of cactus plants. And when you look to see what is happening here, what one finds is that the level of expression of this gene corresponds to the broadness of the beak. So you have a lot of this gene expression, you have a broad beak. You have very little of this gene expression, you have a narrow beak. What gene is this? It's very interesting. It's BMP4. It's the same gene that was killing the cells in the webbing. The same protein that kills the cells of the webbing is used in the face to elongate the beak. So different, and this is a principle of embryonic development which was so important, is that the same protein does different functions in different areas of the body. The same protein which is involved in telling the cells to die in the webbing is telling the cells to divide in the face. Okay, so you can change time of gene expression, place of gene expression, amount of gene expression. Matter of fact, you could show this experimentally. If you add active BMP4, you can actually get a larger, broader beak in chick embryos due to increased mitosis. So this is staining mitotic cells. This is control. This is when you add BMP4. You broaden the beak by adding active BMPs. And the other way of changing gene expression, of getting anatomical change from developmental change, is by changing the gene being expressed. And here we have, this is the gene for ultrabithorax, which is a gene which is a transcription factor gene, and it has a new function in the insects. In the insects, it has this region of polyalanine. This region of amino acids inhibits the distillus gene from being expressed. The distillus gene is one of the fundamental genes that says, make an appendage, make a leg. And so where ultrabithorax is expressed, legs cannot be made. So you have in insects only 6 legs, 3 pairs of legs in the thorax, but you don't have legs in the abdomen. Unlike things like millipedes and lobsters and crabs and shrimp, spiders who have many legs, the insect only has 6. And that's because the legs are confined to the abdomen by this change. So we have these 4 ways of changing gene expression: time, place, amount, and kind. So the turtle. How does the turtle get its shell? Okay, again, we're using a species of turtle which is a pest species. It's the only pest species of turtle. Most turtles are becoming endangered by habitat destruction and so forth, by thermal pollution. Trachomas scripta is the turtle that's found in pet stores. And it is worldwide in its distribution, and it's taking over the habitats of other species. So we don't have any problems using this turtle in our laboratory. It has this carapace, and at the carapace, the bone is covered by these scales, these scutes, and it has a plastron. Okay? Now, if you look at the turtle here, this is one of the Galapagos Island tortoises. You have here the carapace, the bones of the carapace, and the bones of the plastron. Turtle is very strange. Its scapula is covered by, it's within the ribs. It's a strange organism. And the strange thing is made by the ribs. What makes a turtle are its ribs. The ribs go into the skin, and they extend outward, and then they cause the cells in between the ribs to become bone. So there are two main questions here. One is, how do the ribs get into the skin? And second is, how do the ribs tell the skin to become bone? So here we see again, this is looking at the ventral side of the carapace. Here we see the vertebra, and here we see the ribs, and here we see the bone in between the ribs. Okay, so here we see a turtle embryo, stage 16 turtle embryo. This is from Louis Agassiz's book in 1858, the drawing by Sonrel, and the turtle embryo was actually obtained by Henry David Thoreau at Walden Pond while he was doing his philosophy. So here is a special ridge. This is something not found in any other organism. This part of the skin, this bulge, is called the carapacial ridge, and it is the distinguishing mark of a turtle embryo. No other creature has a carapacial ridge so far as we know. And I took this to show you that here's the limb over here. This is not a limb bud. It kind of looks like a limb bud, but actually this is dorsal. This is near the node. This is at the same level as the notochord, carapacial ridge. And here we see a few stages later. This is stage 17. Here is the notochord here. Here is the neural tube over here. And here is the rib, but instead of the rib following the myotome and going down to form a rib cage, the rib is going into the carapacial ridge. It's going into this area of condensed dermis. So this is what makes a turtle a turtle. The ribs go into the skin. How do they do it? Okay, well, we took embryos at different stages, and here we have a turtle embryo at stage 14 where you could just barely see this ridge over here. And here we see it at stage 17 where you could see, here's the carapacial ridge, and here are the ribs going into that ribs. Only turtles have the ribs, have the ridge. Chicks, mice, humans, we don't have this. The ribs go ventrally. So, Chester Intima and Annie Burke were the ones who postulated that what's going on is that the carapacial ridge is secreting a factor which is necessary for the ribs to migrate towards it. That the carapacial ridge is attracting the ribs by putting out some chemical which is diffusing and telling the ribs to go someplace new. And what Intima did was he took out the carapacial ridge of an early turtle embryo and asked, what do the ribs do if you take out the ridge? And what the ribs did is the ribs went into another part of the carapacial ridge. They were still being attracted to a source of something being made by the carapacial ridge. What Annie Burke did was to put metal foil, tantalum foil, in between the rib and the ridge and ask the question, what are the ribs going to do if they don't have the ridge? What they did was they went ventrally just like your rib and my ribs. So it looks like there's something that's attracting the ribs into this area of the skin. So we thought, and actually Annie Burke pointed out, that the carapacial ridge looks just like a limb bud. If I were to give it to people on an exam and just say, what is this? And show a high-power electron, a high-power microscope picture of it, you'd see condensed mesenchyme, you'd see a polarized epithelium. It looks just like a limb bud. So we thought, and so did she, that it would be interesting to look at the genes responsible for limb formation. Okay. And so the first one we looked at was FGF10. FGF10 during limb formation, and it's involved in the mesenchyme, and it tells the apical ectodermal ridge to continue functioning. And so FGF10 is a very important gene in limb development. And we cloned the gene, and we asked if it was found in the same place in turtles as in chicks. And yes, it's found in the mesoderm of the limb, and it's found in the genital tubercle. We grow our turtles at male temperatures. Okay, so it's found in the genital tubercle, it's found in the mesenchyme. Great. Is it found in the carapacial ridge? And the answer was yes. It is found in the carapacial ridge right under this columnar epithelium. You have FGF10 being produced. Good. We were very happy about that. I'm looking back now at this in situ and say we could do so much better, but at that time we were thrilled. Okay, so we have one of the two things. We have FGF10, great. Do we have FGF8? So we tried cloning the FGF8 gene. We had a good candidate. We wanted to know if it was made where chicks make FGF8, in the brain and in the apical ectodermal ridge of the limb. And sure enough, there it is in the apical ectodermal ridge of the limb. We have FGF8, its nucleic acid sequence matched that of chickens almost identically. Great, we have FGF8. Is it in the carapacial ridge? No. Tried some other embryos, younger? No. Tried some other embryos, older? No, it's not in the ridge. We were very disappointed. Okay, so we have other projects to do. We'll put this one aside, go back to it later, some other day. But we couldn't make a model. Then we had a student come into our lab who wanted to work on the turtle project, and we taught her how to do in situ hybridizations. And then we said, okay, let's see if you can do a whole mount in situ hybridization by yourself. We have some old turtle embryos which are beyond the stage we need. We have some FGF8 probe. Show us you can do this. So a few days later, she comes back with her results, and we are astonished. She found FGF8 at the tip of every rib. Not where we thought it would be, but it's in a fascinating place because now we have the place where FGF8 and FGF10 can come together, just like in the limb bud. But it isn't the limb, it isn't the ectoderm and mesoderm interacting. It's the mesenchyme and the limb and the rib that are interacting. So instead of the mesenchyme epithelium interaction, we're having a mesenchyme rib interaction. And this would explain how the rib grows at the same rate as the skin. So we're really happy about that. So what we did is we did something which nobody in their right mind would do. We decided to culture turtle embryos in the incubator by cutting off the heads, cutting off the belly part, and putting them like organ culture, like organs, on a millipore filter in a petri dish. And anyone who's worked on mice or chickens will say, this is a stupid thing to do because you're putting them under hypoxic conditions, and the turtles, organisms need oxygen. And the diffusion we're asking too much for diffusion to work. What we forgot was turtles love hypoxia. They grow under hypoxic conditions. They thrive in hypoxia. You can't kill a turtle by putting it in a CO2 tank. It doesn't work. It'll outlive you. So the experiments actually worked. We put our turtle embryos on filter paper, and we ask them to grow, and they do. Matter of fact, it was kind of bizarre because their tails were moving in the filter, on the filter paper. But what they did was they made a carapacial ridge. And the ribs went into the carapacial ridge, just as one would expect on a real turtle embryo in the egg. Okay, this is great. Then we said, what would happen if we added inhibitors of FGFs to them? If we stopped that FGF signaling between the rib and the ridge, what would happen to the ribs? And what happened was the ridge disappeared and that's where this blood clot is over here, and the ribs went ventrally into the filter paper. So the ribs are going ventrally without the FGF signal. So we now had a model for how the ribs get into the carapacial ridge of the dermis, is that the ribs migrate laterally because they're attracted to a source of FGF10. And once they're attracted to there, they form the rib. Then, so we tested this by adding FGF10 beads to chicken embryos. Okay? We're not going to make them into turtles. We know better than that. But we said, could the rib of chicken embryos be attracted to a source of FGF10? And the answer was yes. That when we added FGF10 beads, you can't really see that in this light, the ribs went to the FGF10 containing beads. If we had saline-coated beads, the ribs ignored them and did their usual thing. But the ribs actually migrated to sources of FGF10. So we now have some hypotheses. Ribs migrate into the carapacial ridges because they're attracted to a source of FGF10, and the outgrowth of the ribs and carapacial ridge can be coordinated by a positive feedback loop between FGF10 in the carapacial ridge and FGF8 in the ribs. We haven't proven that, but it would be a sensible way of doing it. The next question we asked, since we didn't want to look at this question, we asked, what do the ribs do to make a shell? So far we've gotten a novel arrangement of ribs, but we don't have a shell yet. How do you make the rest of the shell? And here we looked at the ribs, and we found that the ribs were doing what ribs are supposed to be doing in vertebrates. They undergo endochondral ossification. They start off as cartilage, and they become bone. So early turtle embryo, the ribs are all cartilage. The ribs then, and this is the same turtle, the ribs then turn their cartilage into bone. Here's the cartilage over here. Here's reabsorption. Here's the bone over here. So cartilage is being turned into bone. And then what we see is along the bone, we see these flares of bone coming. The red here is bone. The blue is cartilage. The red is bone. Here we see bone being formed in between the ribs. And a turtle that's about 3 months old, after hatching, you could see here is the bony plate of the shell. And the shell doesn't completely form for about 2 years. But you could see here is the shell of the turtle growing beneath the scutes, beneath the scales. Okay, so when we did close-ups, it looked like the first set of bones between the ribs actually came from the periosteum itself. It actually was coming from the edge of the bone. But then what happened was, it looked like the cells in between the ribs were being turned into bone. Okay, endochondral ossification. The cartilage produces chemicals such as BMPs. Back to BMPs again. The actual name for BMP, bone morphogenetic protein. That's another thing BMPs do. They form bone. Okay. Cartilage can turn itself into bone. Cartilage can turn into bone by BMPs. But now the rib is encased in skin. It's encased in dermis. Can the dermis respond to BMPs? And the only evidence really that we had was a human disease called fibrodysplasia ossificans progressiva, FOP. It's a horrible disease. It's a lethal disease. It's a disease where people start making ectopic bone, and their body freezes. Their body becomes encased in bone. They eventually die, often of asphyxia, because they can't breathe. So these are all new bones that are here. This is the skeleton of Henry Eastlack in the Mutter Museum in Philadelphia. Some investigators at University of Pennsylvania found that what was causing this was ectopic expression of BMP4. That cells, the lymphocytes, were actually making BMP4, and when they went to a source of infection or trauma, they would release BMP4 as well as antibodies. So this was what was causing the new bone to form, was the connective tissue of the body responding to BMP4. So the BMPs might be responsible for the turtle shell. And of course, when we looked at the amino acid sequence, we couldn't ignore that. There it was. This is telling us we should proceed in this direction. Okay. Now, BMPs, we know that the cartilage is making BMPs. That we don't care about. We know all vertebrate cartilage will turn into bone through BMPs, or the rib cartilage makes BMPs. What we're interested in is the skin responding to BMPs? And for that, we use an antibody to phosphorylated SMAD1. BMPs will phosphorylate SMAD1 and SMAD4. And so we want an antibody that only bind to phosphorylated SMAD1, not to SMAD1 without phosphorylation. We got an antibody from Dr. Heldon which did this, and what was found was that the cells of the skin were positive for phosphorylated SMAD1. That here are the skin cells around the bone. The blue is bone here. And what you see right next to the bone are positive cells, cells that are positive for phosphorylated SMAD1. And you see this little tongue over here, this area of SMAD1 responsive cells. You go a few slides later and you find that it's bone, that this is the edge of the bone that we're looking at here. And so we kept on finding that phosphorylated SMAD1 comes right before bone formation. So here is looking at the cartilage and the bone forming. And here we see this group of cells that are being reoriented in the skin. And what we then find is that these reoriented cells are phosphorylated, have phosphorylated SMAD1. They are responding to BMPs. I just took a lot of pictures that day because it was just so beautiful and all the conditions were right. Here again, here are the cells responding to BMPs that are put out by the cartilage and becoming bone. So it looks like FGF10 is secreted by the carapacial ridge dermis. The ribs migrate into the carapacial ridges because they're attracted to the source of FGF10. The outgrowth of the ribs and carapacial ridges grow together because of this feedback. And each rib serves as a signaling center for the formation of the costal bones of the carapace and the growth of the bone from the rib periosteum outward. Okay, so each rib acts to organize the cells around it to become bone, and it looks like a major signal for costal bone formation is a bone morphogenetic protein that causes the dermis to ossify around the rib as the rib undergoes endochondral ossification. Back to the Creation magazine. Okay, the title of the paper by Leeson and Gilbert summed up the evolutionary conflict perfectly. Okay, this is the same slide, but I want to say, paper by Leeson and Gilbert. Tyler Leeson comes out in 2013 with a theory of how the turtle got its shell. But it's not coming from developmental biology. It's coming from paleontology. Leeson found that Untilosaurus, a lizard of about 260 million years ago, has ribs that look like developing turtle ribs. They're flattened out. It looks like there's some turning of skin into bone. Not only that, and this was incredible, when you look to see if there was muscle attachments for the intercostal bones of the rib, like you have in our ribs, like spare ribs, you know, you have muscle around each rib. Turtles don't have that because their ribs are in the skin. If you look at Untilosaurus and you look at the fossils, you find that there's no evidence of intercostal muscle attachments in their ribs, except in the two places where turtles have them, the back of the back rib and the front of the front rib, because that's where the muscles attach to go around the entire respiratory system. So it looks like Unitosaurus is the ancestor of the turtles, and it fits perfectly with our developmental biology scheme. And he actually says here, Unitosaurus ribs developed like turtle ribs and only like turtle ribs. The growth of the bone outward from the rib periosteum, the T-shaped broadening of the ribs as in turtles, shortening of the trunk to 9 dorsal vertebrae, no insertion points for the intercostal muscles like turtles. And they talk about this and they call it actually the evolutionary developmental model for the origin of the turtle shell. And by looking at the phylogeny of here, they show actually that the only thing that it looks like, the only thing it resembles, are turtle ribs. And so we're very happy to say that, okay, they may point out that a year earlier that we didn't know what the ancestor of the turtle was. Thanks to the work in developmental biology and paleontology, we now do. Okay, I should also mention that a paper came out last month by Leeson and Beaver, Gautier, which actually looked at the skull of Unitosaurus and said it is developing like a turtle skull. So it looks like the whole picture is coming to completion. It looks like we finally have found the ancestor of turtles. It's a diapsid ancestor, which the museum people were not happy about because there was a battle between the molecular people and the museum people. But here was a museum specimen which showed that it was diapsid, so I think everybody is going to be happy. But it's, I think, a very important finding. So we have how the turtle gets its carapace shell by changing the expression of FGF10, which changes the expression, which changes the direction of rib migration, which now in the skin can organize the shell around the rib. We can go back to Thomas Huxley. Evolution is not a speculation but a fact, and it takes place by epigenesis. To understand evolution, one needs to understand development and how it can be changed. Turtles live dangerous lives. Turtles are risk takers. But what David Quammen said I think is wonderful. Nature interests me because it is beautiful, complex, and robust. Evolutionary theory interests me because it explains why nature is beautiful, complex, and robust. And I just want to give a shout out to all the people who have helped us and supported us, the National Science Foundation, Swarthmore College Faculty Research, the Howard Hughes Medical Institute, the Academy of Finland. Most of this work was done by undergraduates in our laboratory in association with Judy Sieber Thomas at Millersville University and Rocky Twan at Thomas Jefferson University. And so I just want to say that it's wonderful being here and talking about how newness can come into the world, since my field, evolutionary developmental biology, really started with some observations of classical embryology done at the borderline of invertebrates and vertebrates. It started off as a Russian science, and it's just wonderful to be here talking about it to you. Thank you very much.
First of all, let me thank you for your wonderful lecture. It was really like a detective story for me. And I want to, if you have any questions, just do not hesitate to ask our lecturer. And I'm going to translate in case you need my assistance. Если вам понадобится помощь при переводе, то прошу задавать вопросы. Я собираюсь их переводить с английского на русский и с русского на английский язык. Если вы хотите задавать вопросы по-английски, то это всячески. Татьяна, встаньте, пожалуйста, поднимите руку, кто хочет задать вопрос, и давайте. Ну, здесь не думаю, что понадобится язык.
We have a slide about humans and chimpanzees. So my question is it possible to grow a human being from a chimpanzee? just manipulating just two genes?
Yeah. The answer to that is almost definitely a no. First, what we know about are the genes that are involved in controlling mitosis. We don't know about the genes that are controlling neural cell migration, which is also very important in human brain formation. And of course, what makes a human also are, some of the body parts involved, the opposable thumb and all sorts of other things, musculature. So we can't get a human being from a chimpanzee. We might be able to get a chimpanzee with an abnormally large brain, but without the cell migration, it's not going to amount to anything much. How the cells know when to migrate and when to get off to form the cortex is another critically important human thing. We don't know that yet.
Okay.
Hello. Hello?
Hello.
Yes. The question is, you said that the FGF8 and FGF10 were the most important in finding why did ribs were attracted to the carapacial ridge. So the question is, why did you decided to find the place of expression of those two genes, except for the superficial resemblance of the carapacial ridge to the limb?
Yep. We looked at those in the first place to see if they were there. Then we looked at other FGFs as well, and we saw them, but in very small amounts. It looked like these were the major ones. And so we tried some inhibitory experiments blocking FGF receptor 2 and 2A and 2B and so forth. They didn't amount to much. So we're saying that it's an FGF, probably 10, probably 8, but it could be other members of the family that bind to the same receptor. So we're saying FGFs. We're also saying BMPs. We know that SMAD1 can be phosphorylated by several BMPs. We know that BMP4 works, but that doesn't mean that BMP7 doesn't work. Yeah, okay.
Dear Professor, have you some hypothesis about placodont shell occurrence?
The placodont?
Placodonts, convergence situation.
Okay, the placodont shell.
Yeah,
okay.
Yeah.
We do have a hypothesis, and that's the second part of the talk, which is I told you about the carapace. I did not tell you about the plastron. The plastron, we think, and I'm talking heresy, the plastron we think is made by neural crest cells, trunk neural crest cells. Now it says in my book that trunk neural crest cells don't form bone. We think in the turtle they do. The cranial neural crest cells form the facial bone. The trunk neural crest cells form nerves, they form glial cells, they form pigment cells, they form the adrenal medullary cells, but they don't form bone or cartilage. We think that in the turtle, there is a second set of neural crest cells that migrates ventrally. And this second set of neural crest cells is respecified as cranial crest. Even though they're coming from the trunk, we think that they are active as cranial crest cells and have the capability of forming bone. Now, Brian Hall and others have claimed that the placoderm, that those bony fish, those primitive fishes, the placoderms, have bone that look like it's neural crest derived. And so they're saying that most of the vertebrates, after the placoderms, lost the ability to have bone coming from the crest. Oh, I'm sorry. I'm sorry.
Placodons, which related with plesiosaurs. Reptiles, not fish.
Oh, I'm sorry. I was thinking of the bone. I was thinking of the fish. Oh, okay. Okay. No, I'm sorry. The placodonts being which group of? Can you give me an example?
Closely related with plesiosaurs, plesiosaurs, and ichthyosaurs.
Yes. Okay. I'm sorry. Yeah. Okay. Still, I can go on the same path because the placoderms were the fish. Okay. The placodonts, such as the plesiosaurs, mosasaurs, whatever. Okay. Those have gastralia. These abdominal ribs. Alligators have gastralia. Turtles have a plastron, and it looks like the plastron is derived from gastralia. It looks like the mosasaurs, the plesiosaurs, the T-Rex, those animals with gastralia. Turtles also have gastralia, but they've been modified so they don't look segmental anymore. And what's really interesting is that the first turtle we know of, Papacalis, which is about 250 million years ago, has a plastron that looks like gastralia. So we think that those, I'm sorry for the placodon mixture, but they come from, I think they both come from the neural crest. Yeah.
Thank you.
Okay.
Hello. Hello, I also have a question for you, and I didn't understand, or perhaps I missed something. Could you tell me, please, about why did you explore these genes, why did you explore these genes, and how did you understand that these genes are necessary to explore their expression, etc., and what is known about their origin?
Okay. Okay. We looked at dozens of genes. These are the ones that worked. Okay, so I could show you expression patterns of sonic hedgehog, MSX2, all sorts of paracrine factors, BMP7, a whole gamut of paracrine factors. And those weren't in the place where we wanted them to be at the time we wanted them to be. So, but they were in interesting places. What we found was all sorts of other things that I haven't told you here, like the sonic hedgehog pattern marked out every scute, every scale on the back of the turtle has a dot of sonic hedgehog on it. It's really beautiful. But it had nothing to do with forming the shell. It has to do with forming the scutes on the shell. So we did look at others. What really caused us to look at these were the suggestions of Annie Burke and our own observations that this really looked like a limb bud, and we thought that the FGFs would be the best candidates. Now, another set of candidates would be the Wnt proteins, and we actually found that Wnt is active here. Wnt3A seems to be active, and beta-catenin is accumulating in the carapace ridge cells, as you would expect, not only in other things, but also in the limb bud. So the Wnts are probably active too. We haven't looked at the Wnts. We've looked at the hedgehogs, the BMPs, the FGFs. And we looked at beta-catenin to see if it was accumulating in the carapacial ridge. But these are the ones that worked. There were several that didn't.
Hello. You said that FGF10 attracts chicken ribs.
Yes.
So do chicken ribs express FGF8 on the tip of it?
We haven't looked at that.
If it is expressed in chicken ribs, then your second hypothesis is right. So
yeah, we have not looked at whether FGF8 is found in chicken ribs. We could. We could very easily do that. We have lots of chicken embryos.
Okay.
But we haven't. No, thank you.
Thank you for the answer.
No, we haven't done that.
Hello? It's working. You talked about the carapax and then answering the question about the plastron. And what about the way they become connected?
Yes. That is what we are working on at the present. Because we found some fascinating things, and we, on another, I just got a movie on email, which from the researcher that I'm working with in Helsinki, showing the earliest plastron bones. And what you see in the very early stages of the plastron is the plastron, two of the plastron bones on each side send a process up into the carapace. And when they reach the carapace, they go right next to ribs, but they don't join the ribs. It looks like they are waiting for the ribs to make bone that they can connect to. They don't connect directly to the ribs, which surprised us. We thought that there was a definite connection. So we're just now looking to see how the sutures are made there. And my colleague, Ritva Rice at University of Helsinki, is looking at the sutures of the plastron because they look just like the sutures in the face and skull. They look like they have the same molecular biology. They look like they have the same structure. And I could show you all sorts of pictures about this. She is most interested, though, in this rare thing, these bridges, these coming up from the plastron bones, and, well, they look like jaws. They look like they're coming up like jaws. And so we joke that the turtle is the beast with two faces, that the turtle plastron is like a second face. It's made of neural crest cells, and here's this bridge coming up, which looks like the bridge to a jaw. So, but that's just, analogy. That's strictly analogy and perhaps a joke. So, but the bridge, the bridge does not arrive from the carapace. It arises from plastron cells, which send processes dorsally. Yeah.
Any other questions?
Michael Rosanov, Pirogov Medical University. Could you possibly tell a few words about new terrains, the most sizzling, exciting growth points in developmental biology? Well, I know, you may need a lecture for that, but just a few words.
The growth points of developmental biology. Let me give you three. One is the brain. That's kind of a no-brainer. I mean, it's the brain, the origins of the brain cells, the migration of the brain cells, the coordinated growth of the brain cells is a new horizon that the technologies are finally becoming able to look at. So that's one area. The second area is plant developmental biology. I think that's where the real interesting action is right now, is in plant growth and development. I think plants are going to not only make our food, they'll make our building materials, they make our fuel. I think that changing the development of plants is going to be a fascinating area. I think the third really big growth area is developmental symbiosis. That the fact that we are not only created by the cells of the fertilized egg, we are also created by bacteria. And the bacteria are not only in our guts to eat our food with us and to make some vitamins, the bacteria help make our gut capillaries and help make our gut-associated immune tissues. Without the bacteria, mice have a very poor capillary network. They have very poor immune sites. Matter of fact, they have an immunodeficiency disease, and also they have behavioral anomalies. The bacteria are inducing gene expression in our cells, and the gene expression is normal gene expression. We expect the bacteria. They are part of our developmental program. They are part of how we become who we are. And I think that looking at us not as an organism which gets bacteria, but as what's called a holobiont, the organism plus its persistent symbionts is going to be a critically new way of looking at life. For instance, mother's milk has two sets of nutrients. One is for the newborn baby. Fine, that's accepted. There's a second set of nutrients in mother's milk that the baby cannot use. They're not for the baby. They're special sugars for bifidobacteria, one of the good bacteria that we want in the gut of our newborn. So bifidobacteria have 7 enzymes, has genes to encode 7 enzymes which specifically degrade these sugars into metabolizable glucose. The baby can't do this. This is something, the symbiont, this is made for the growth of the symbionts. And so we usually think of birth as the birth of an individual, but it's also the maintenance of a community. And so I think looking at organisms as complex ecosystems is another area of developmental biology, which is going to be very big in the very near future.
Hello?
Yes.
Could you please explain what for did you need to cut off the heads of turtle embryos?
Mm-hmm. Yeah, because they wouldn't fit on the filter. And so that was the main reason, was they wouldn't fit on the filter. And also, we were just wanting to look at the growth of the shell.
Okay.
You know, and so the head was really, I'm sorry to say, getting in the way. And so the head was cut off to do the experiments.
Okay.
Yeah.
Thank you.
Yeah.
Hello. I have a funny question. Can turtle grow without shell to adult one?
Okay. Can turtles grow without shells? No. It depends. What do you mean by the shell? In that, if you mean can they grow without the entire shell, yes. And there are some species of turtle, like the leatherback turtle, which has the ribs going into the skin, but they do not make the costal bones between the ribs. So they don't have what we would call a turtle shell, but they have the beginnings of the shell. And we think that the dermis of the leatherback turtle is very thick and can be protective in its own right, and it probably contains a lot of BMP inhibitors, like gremlin, which I showed earlier, to stop the ossification from happening. And so you can get a turtle without the full shell. There are other species of turtles, sometimes called the pancake turtles or the soft shell turtles, which have part of the shell. They have the first part of the carapace, the ribs are into the dermis, and then there's a small amount of ossification near the spine, but it doesn't go outward. So they only have part of a shell. So you can get a turtle without having the complete shell, but the turtle really becomes the turtle by putting its ribs into the skin, and anything which does that, the only thing that we know that does that so far are turtles. Thank you.
Good evening. Good evening. As far as I could understand, you were working with chicken and ducks, and turtles as your main object of investigation. But my question is, are you going to work with other objects, and what gene expression are you going to look for and investigate?
Okay.
Thank you.
We're looking right now at the genes that might be involved in forming the plastron. And we're looking, one of the main projects we have right now is to do transcriptomes of the plastron cells, transcriptomes of the early neural crest cells, and transcriptomes of something that I hadn't mentioned earlier, a late migrating population of neural crest cells, which we think forms the plastron. We want to look at especially Hox gene expression, because Hox genes are important in stopping the neural crest from forming bone. In culture, Arhad Abjamov has shown that if you put trunk neural crest cells in culture, they eventually lose Hox gene expression, and when they lose Hox gene expression and you put them into micromass culture, they'll form bone and cartilage. And so one of the things we're looking at is a series of genes which should be activated in the cranial neural crest, which are not active in the trunk neural crest. Another one is PDGFR alpha, the alpha subunit of the receptor for platelet-derived growth factor. This gene is found in head neural crest cells, in the cranial neural crest cells, not in the trunk neural crest cells. We want to see if it's expressed in that new wave of neural crest cells that is capable of forming bone. Now, in terms of other organisms, Dupuy at University of Helsinki is starting to look at a group of reptiles, including reptiles which are called, flying dragons, which may have ribs going into their dermis. Nobody has looked to see if this kind of thing, which we thought is turtle-specific, may be found in other organisms. And so he's looking at a series of reptiles that we haven't been looking at, but we're keeping in touch with them.
Thank you very much.
You're welcome.
Just a second. Take the microphone.
Is there any difference in embryonic development between Trionyx and other turtles?
Yes. Yeah. The difference in development. Yeah. The question. What?
Then what is Kreatsa, big turtle, skinned turtle, not sea turtle?
Yeah, not sea turtles. No, we're talking about the soft shell turtles, Trionyx. Yeah. The soft shell turtles differ, I think, enormously in development from Trachomus, which is our model turtle. Now, Shigeru Kuretani has been looking at Pelodiscus, which is a Trionyx type turtle. And what he finds is that the carapacial ridge is not as important after it initiates rib migration. Then the carapacial ridge migrates faster than the ribs do. And so the ribs stay in a very small area where the skin goes in a much larger area. That means that the ribs don't meet the carapacial ridge. And when the ribs don't meet the carapacial ridge, something very interesting doesn't happen. Okay? When the ribs meet the ridge, it stabilizes BMP expression and sonic expression in the carapacial ridge. And BMP and sonic are together in the ridge. And those people here who know about hair follicles and teeth development and feathers know that when you put BMP2 and sonic together, that's when you get epidermal placodes. And what happens in Trachomus is you get the beginnings of epidermal placodes at the carapacial ridge, and by reaction diffusion, you get all the spots where the scales are going to form. And so Trionic type of turtles don't have scales. And so not having the ribs meet the ridge not only puts the skin at a distance from the ribs, but it stops the scutes from forming. And so we've done a number of experiments with both Trionyx and Trachomus, and Trionyx looks like an early stage development of trachemus carapacial ridge. Welcome.
Mr. Gilbert, the first in the row of persons you have shown Suzumi's Ono.
Yes.
Maybe meaning evolution by gene duplication.
Yes.
But you till now haven't said anything about its role in development.
Right.
I mean lateral duplication, amplification,
okay.
or polyploidization,
okay.
and tandem.
Okay, okay. The turtle genome has just been published very recently, and we don't know much about gene duplications. I didn't mention Susumu Ono's work because we haven't done anything using it, but I did mention that one of the genes that controls mitosis is SRG A, and this SRG protein, SRGAP2A, is important for cell division in the brain. But by gene duplication, you make 3 more copies of it. One of those copies is a faulty copy. It inhibits the original copy. And that's the inhibitor that humans have evolved by gene duplication. We've evolved an inhibitor of the original gene, and that's partly responsible for brain growth. So I did mention something relating to Ono's work, but it didn't fit into the turtle story. Thank you.
Hi. I have a question about turtle life. You know, I've heard a story that turtle shell grows not as fast as turtle does, and at one moment the shell is getting not so big as turtle, and she can't breathe and she died. So can you tell me about the main reason of turtle death? And another question, can we do something with its genes, so do something that shell will grow bigger and bigger and bigger to make the turtle immortal organism or something like that?
Okay. I haven't heard that story of turtle death, though. The turtle growth that I've seen has been coordinate with the body. I don't think turtles die by overgrowing their shell. Matter of fact, if you take a snapping turtle, it looks like it's constrained in a girdle. It looks like it's much bigger than its shell already when it's born, which may be why they're so nasty. But I don't think that turtles die because of miscoordination of growth. Turtles die from infection, and they die of trauma. They die of being eaten by raccoons and other animals. The turtle physiology is absolutely fascinating, and no one, to my knowledge, has studied turtle stem cells. Mesenchymal stem cells of turtles, I think, will be very interesting. But a 40-year-old female turtle lays more eggs than a 20-year-old female turtle. That they do not have your normally expected decrease in physiology over age. The immune system of a 40-year-old turtle is every bit as good as the immune system of a 20-year-old turtle or a 3-year-old turtle. Turtles have probably the slowest metabolism that you can actually stay alive with. They just keep their metabolism at a very, very slow level. They can hibernate for decades, which is probably one of the reasons that they survived, the KT extinction. The dinosaurs got knocked out and whatnot. The turtles probably slept through the whole thing. They can really slow down their metabolism for decades. And so they also have a remarkably high level of reactive oxygen radical scavengers, so that the death of tissues and the death of proteins due to reactive oxygen species is minimalized in turtle embryos and in turtles themselves, in adult turtles. So the physiology of turtles has a lot to commend its study. When people did the genome of the turtle, they thought, oh, we're going to find out how the turtle, produces all these reactive oxygen species inhibitors and how they, survive hypoxia and how they do this and how they do that. Nothing's come out of the genome yet about that, I'm sorry to say. We expected a lot. We did not get anything else out yet except the normally expected genes. But I think that now that we do have a turtle genome and we can look at how the genes are activated, we might be able to find out how the turtle survives hypoxia, how it makes reactive oxygen scavengers, and so forth. Right now, we don't know.
Sorry, but I don't speak English very well.
Better than my Russian.
The question is in Russian. Скажи, а можно подробнее объяснить механизм дыхания черепах, как в связи с наличием панциря?
So she asked whether you can explain in greater detail the mechanism of breathing of turtles connected or in relation with its shell.
Yeah, okay. The breathing of turtles in connection with its shell is very interesting, and they have a musculature like a diaphragm. Since they don't have a rib cage, they have a new set of muscles. We actually want to find out where these muscles are coming from. I think that they are the intercostal muscles, but we haven't been able to show that yet. I think that there might be a respecification of the dermamyotome of turtles to make more dermis, less muscles, but those muscles that are made may be the ones that go around the lung. But one of the things about having a muscular instead of a bony cage around the lungs is that the turtles have to coordinate their breathing with their motion. And that's why turtles can't sprint, can't run long distances, because they have to coordinate breathing with locomotion. And that's not an easy thing for a turtle to do.
Shall I translate? Yes? No? Okay. Any other questions? Probably. Oh, I think it's enough, and I will give the microphone to the organizers. In the end, I would like to say that this is a great pleasure to have Scott Gilbert here, and I would like to thank everybody who took part into organization of the visit. And first of all, I would like to thank for financial support. First, USA Embassy, the program, the US Speakers Program, and All Russia Festival of Sciences, and also all people from the departments of evolutionary biology, embryology, and invertebrate zoology, Institute for Developmental Biology, and also our translators, Anastasia, she's from courses English for Biologists. Actually, she's a philologist. And last thing I would like to say that we will have welcome party at the Department of Invertebrate Zoology, and the room number 594, 594 аудитория. And if you like to ask some more questions, we will provide tea, coffee, and cakes, so you are welcome. And probably some final remarks. We would be greatly honored if you sign your book that we found in our library, the Scientific Library of Moscow State University. And if you sign it, we would be really greatly honored, because students will read the book not taken from the library with your sign. And it would be...
Thank you all very much, and thank the organizers, Julia, especially you, Dr. Julia Kraus. Thank you so much.
And you see the queue.